So the next talk is from Umberto Romeo from CordenPharma.
Okay. Maybe I need this one, right?
Yeah.
All right. Can you hear me well? I guess. So I'm actually heading the development organization of one of our sites that is heavily involved in nanocarriers development. And today I would like, let's say, to share with you what we're doing in our company and some innovation aspects that we're trying to introduce. Just a few words about CordenPharma and what we're doing. So actually we're a global CDMO. We are supporting companies in developing and manufacturing different type of active ingredients, excipients, and as well drug products. And we are slowly moving also in advanced medical therapies, including nanocarriers actually. We are roughly 3,000, globally polydispersed, I would say. And we have almost, let's say, 1 billion in revenues counted last year. There are 12 facilities, mainly present in Europe, and we have 2 facilities in the US. We are owned, of course, by a private equity, and the company is start aging, so it has been formed in 2016. Our, let's say, business model is actually divided in platforms. So we have different business units. We have actually 5 and a newborn that is bringing the count to 6. The newborn is related to the oligo platform, while of course we are active in different fields, including peptides, lipid and carbohydrates, injectables, that includes also nanocarriers, and of course small molecules and high potent drugs. And this is just to let you know where we are, if you want to visit us, of course. Today, of course, I would like to discuss a little bit something more focused on nanocarriers. Of course, this slide might be obsolete, given the fact that most of you know a lot about lipid nanoparticles. But I like, let's say, to define lipid nanoparticles as 2.0 liposomes. So they are a very specialized form of liposomes designed to carry nucleic acid. And the magic ingredients is represented by the ionizable lipid or the cationic lipid, right? So this lipid, in fact, interacts with a negatively charged nucleic acid, starting creating, of course, the ionic bond that will definitely trigger the self-assembly mechanism. Most of the newest way to manufacture this type of particles are based by an evolution of what we call ethanol injection. And that consists, of course, in dissolving the lipid formulation into absolute ethanol and mixing it rapidly with an aqueous phase containing our cargo. The results, it's actually the self-assembly of a nanoparticle, right? So we started to run some internal innovation program, even if we are not a discovery company at all, given the fact that we have a nice integrated business with our lipid platform. And the idea was to start screening our lipids with also, third-party lipids in creating new lipid nanoparticle composition and to assess what actually is the real difference in their transfection efficacy. So we started doing transfection efficacy on immortalized human cell lines, and primarily we focus on egg cells. But of course, the idea is to expand further. The primary endpoints of these initial studies were, of course, determining the transfection efficacy and, of course, determining a little bit also some structural features of these nanoparticles between the different formulations. The cargo molecules that we identified to manufacture this nanoparticle is standard GFP. Of course, it's a mature mRNA with a Cap2 and a poly-A tail. So in this diagram, I'm just simplifying a little bit the way that we used to manufacture actually the nanoparticles. So of course, we decided to go very small scale, so at ml scale. The organic phase, of course, was done in an Eppendorf tube using absolute ethanol, and of course, we prepared an aqueous buffer, primarily citrate, right, at pH 3. We have different technologies to make nanoparticles also in the lab, but we went with the ones that enables us to manufacture the smallest amount of nanoparticles, also to spare a little bit of very expensive mRNA. And we actually moved with a cheap technology. It's a tongue twister, it's called MHF, so microfluidic hydrodynamic focusing, that enables us actually to manufacture small scale nanoparticles. The nanoparticles were then, of course, diluted and purified through centrifugal filtration by Amicon filters. So It's a quite complex slide, but just to show you all the combination that we started testing in order to determine different type of potential formulation. Some of these formulations have been done with our lipid starter kits that you can see in the picture, and that they are actually designed to simplify actually small scale studies and the manufacture of lipid nanoparticles at lab scale. We used, of course, different type of neutral phospholipids. We decided also to try out phosphatidylserine, that is a negatively charged lipid that seems quite interesting in terms of enhancing transfection efficacy. We use different ionizable cationic lipids that include C12-200, DOTAP chloride, and CKK-E12. And of course, we used also different type of cholesterol, including our vegetable source cholesterol and different stealth lipids as well. So the result was quite interesting. In any case, we had to change and play a little bit with the process parameters because, of course, different composition definitely require different processing parameters. So we had to modify slightly for the different formulation the concentration of the lipid mix between 12.5 and 25 millimolar. We had to also use a citrate buffer at pH 3, and the aqueous to organic ratio had to be modified for certain formulation. Some have been done with 6:1 ratio, the other with 3:1. The total flow rate with this system is 12 ml per minute, and all the formulations have been diluted 1:2 in PBS, in line actually. So we selected here some winners. Our, of course, selection was done considering some important physical characteristics of the particles. So we decided to keep the formulation with, of course, a size smaller than 100 nanometers post-purification, and of course with a PDI below 0.2. And actually we lost 2 formulations on the road. Very likely that formulation required a little bit more of fine-tuning. But actually, we can say that already with the first iteration, we get quite good results in terms of physicochemical characterizations. Also, the concentration of the nanoparticles have been measured by moles, and actually, we have a quite high number of particles in all the formulation, having the 4 and the 37 yielding higher than the other ones. Of course, we also verified that they were not empty particles, right? So we performed a ribogreen assay, determining also encapsulation efficiency, and actually almost all the formulation had a very good and nice high encapsulation efficiency. The only 2 that unfortunately didn't make it to the final round of testing were LMP 31 and 32. Unfortunately, the ones containing as well also phosphatidylserine, so the idea is to remake them in a next iteration. And the winning formulation have been submitted also to cryo-EM for characterization. It's not that easy taking picture of nanoparticles, but as you can appreciate, there are structural differences. Especially LMP4 and LMP36 show a very nice crown or corona. With a slightly electron-dense core, while the other compositions have more multilamellar shaped organization. We decided, therefore, to do a small benchmark for transfection efficacy, and we decided to select 2 combinations that are very well known. So these combinations have been identified with LMP1 and LMP2 that are actually the Pfizer-BioNTech formulation and the Aquitas formulation, and the Moderna formulation, right? So the LMP1 contains, in fact, the ALC0315 and, as speculated lipid, the LC0159, while the LMP2 contains the famous SM102 ionizable lipid. And then the third formulation is, let's say, made-up formulation containing a very well-known ionizable lipid that is the C12-200. So we use this formulation, of course, characterize them again to be sure that they were, let's say, good. And what we find out is that, of course, in the second iteration, the size changed a little bit. We had LMP1 and LMP2 that are very small, yielding roughly between 60 nanometers and 80 nanometers, while the LMP3 is slightly larger, while the PDI, of course, is quite outstanding and almost close to 0, especially for LMP3. After purification, we get a slight increase in size as expected, as we are, in any case, compressing the particles against a membrane. But in any case, let's say all the formulations were quite valid, and also the encapsulation efficiency was quite good. The pictures again are confirming the structure, and it's also surprising how cryo-EM confirms, but actually the size of the particles of the formulation 3 are slightly smaller from the one predicted by the DLS. So I'm really happy that there are also other tools like the ones that we saw just in the previous presentation to confirm size as well. And therefore, we decided, let's say, to use this formulation in a transfection study. The transfection study protocol foresee the use of a hexagon line. We actually incubated the cells for 3 hours with a 20% of FBS buffer, apologies, and the cells have been incubated for 21 hours. The positive control in this case was a slightly suboptimal positive control because we used actually electroporation using naked RNA to transfect the cells, and of course that yielded a slight higher signal at flow cytometry. In fact, we used as a readout method flow cytometry given the fact that we were using GFP, right? So these actually are the results that are quite interesting. So what definitely we can say is that the Moderna and the biotech formulation work as expected. And, but the most surprising fact is that in any case, the LMP3 formulation is definitely comparable or even slightly providing a higher signal, at least in terms of transfection efficacy in this specific cell line. So the result was quite reassuring. So our idea now is to move for next steps in additional characterization of other composition, including the ones that we showed in the slide, and eventually even investigate the possibility to use new lipids that are not, let's say, available out in the market, and perform this in vitro screening not only in immortalized cell line, but also in primary cell line, determining mRNA expression by quantitative PCR, and trying to study a little bit more the endocytosis mechanism. That is a slight, large incognita still in let's say, the academic world. And the idea is at the end to identify the most promising formulation to be brought, I would say, in vivo proof of concept studies with the right partner, right? So thank you very much for the attention, and of course, at the end of the session, I'm open for questions.
Thank you. So I'm the next speaker on the list. First of all, I would like to thank the organizer for the invitation. And so today I'm going to focus on what the two companies are doing, so Adigen and Diving Cells. Just before starting, just a bit of history about the two companies. This one is moving on its own. So Adigen was founded by Neil Desai about 10 years ago. Then we decided to found Diving Cells. And Diving Cells was founded in south of France. And we, at Diving Cells, we are doing mainly all the R&D study of this approach. So what we are developing is a peptide-based nanoparticle for gene modulation in cancers. We have developed this, what we call ADGN. It's a platform based on peptide for nucleic acid delivery. And it's mainly, can we have it? Not moving. It's mainly based on amphipathic peptide, which combine electrostatic interaction and hydrophobic interactions. And so this amphipathic peptide can interact with oligonucleotide. We can interact with different sizes of oligonucleotide. And we also have the possibility to add a targeting motif at the end terminus of the peptide. And I'm going to show you, we had some tumor targeting motif. So this peptide interact with oligonucleotide through a self-assembly mechanism, and they form nanoparticles. So we have first electrostatic interaction, then hydrophobic interactions. It's just going back. And so we have particle size of 100 nanometers. So Adigen peptide forms stable nanoparticle with different type of oligonucleotide. The size is around 100 nanometers diameters. The charge is neutral, plus minus 10 millivolt. We reach 100% encapsulation efficiency. We show that the particle can be stable as lyophilized powder. we demonstrate that we can protect DNA, RNA for degradations, and we increase the stability of RNA in vivo. So this novel ADGN approach is for gene modulations. Is it a way this is not going to go on its own? So this, what we have developed is a system for systemic delivery of mRNA. So we can use the ADGN nanoparticle for mRNA rescue, mRNA replacement, just to bring wild-type protein, wild-type mRNA in the cells. Or we can also use this for gene editing. In that case, we get the CRISPR machinery inside the cells. Just a couple of examples. Our thing is to do IV injections. So that example shows that we did single IV injections in mice of a nanoparticle containing mRNA expressing luciferase. And we got a strong labeling of the tumor with the luciferase, strong expressions. Is there a way stopping it moving? I don't know. And the second example is to deliver CRISPR machinery. What we're doing here is we have an orthotopic lung tumor, and we implant the tumor in the mice, and then we treat, this tumor is expressing luciferase, and we treat by a single injection of the nanoparticle containing CRISPR machinery. And what we can see is basically we can completely edit the luciferase inside the mice. This is 2 mice models there. So our strategy, one of our main projects is to use this ADGN technology to target one of the major oncogenes, is the KRAS oncogene. We all know that it's one of the major players in cancers. KRAS is mutated in 48% of the cancers, and the interesting thing is 72% of the KRAS mutation are located at yeah, can we just... Yeah, the mutation is only on one codon, mainly on one codon. It's a G12 codon. And if we analyze in detail, this G12 mutation is mainly the major substitution at G12D and G12V, and to a minor extent, the G12C. So far, there's no drug on the market approved which are able to target G12D and G12V mutations. Just recently came out 2 small molecules, sotorasib and agrabasib, which target G12C mutations. There's a way just to pass slide by slide. And those 2 small molecules, sotorasib and agrabasib, have been approved by the FDA, and they are efficient in clinic, but it's okay? Yeah. So sotorasib and agrabasib target G12C mutations. They are active in clinic, but they are limited by the rapid emergence of resistance. So there's still a need to have a molecule to target G12C. So what we have developed at Diving Cell is the ADGN technology. This technology is to direct target KRAS mutation using the CRISPR machinery at the codon 12. Our strategy is to permanent edit those KRAS mutants. We have 3 formulations called ADGN 121, 122, 123. Those formulations contain the gene editing machinery, mRNA encoding for Cas9, and a small guide RNA, which is specific for each mutations. And all those mRNA are in our nanoparticle, peptide-based nanoparticle, with the advantage of being able to target the tumor. So we first have validated those 3 formulations on cell culture, and we show that on the left side that in the correct background, for example, ADGN21, which is targeting the G12D mutations, in the PONC1 cells, which is G12D mutated on KRAS, get more than 80% efficiency in terms of gene editing. And 123 is 80% gene editing in the correct background in the SW403 cells. and the 122 is also around 80% gene editing in the correct background in cancer cells with G12C mutations. Gene editing of KRAS leads directly to a reduction of cell proliferation, as you can see on the other graph. For example, the 121 block PONC1 proliferations, 123 SW403, and 122 block the H358 proliferations. And it's highly specific, there's no cross reactions. So we next have analyzed the different formulations in vivo. And first we analyze the efficacy of 121 on the KRAS G12D background. No, it's still moving. On the G12D background, for that experiment, what we did xenograph tumors. We used the human pancreatic carcinoma cells, which are having the G12D mutations. And we did only 2 injections of the 121 at day 1 and day 7, and then we look for tumor growth. And as you can see here, we have the 2 control. One is a normal control, saline buffer. The second control is using the nanoparticle with a non-specific CRISPR complex. And then we have a dose response with a ADGN 121. At our 1 mg per kg, we completely abolish the tumor growth, and we even see if you can see the central graph, which is a waterfall plot, we can see 80% reduction of the tumor volumes. So there's high specificity, no cross reactions, and the treatment was very well tolerated. We did a deep analysis of the tumor, and we look inside the tumor what's happened in terms of the level of KRAS G12D, and we find that in case of where we are using 1 mg per kg, the group 4, we have an editing of more than 80% of G12D, so it's directly editing KRAS G12D and also blocking the downstream pathway. We then analyze the efficacy of ADGN 123. It's in a KRAS G12V background in vivo. So we use mice injected with human colorectal cancer cells, which have G12V mutations. As previously, we did only 2 injections of the nanoparticle at day 1 and day 7, and then we look for tumor growth. And as previously shown with the ADGN 121, we can, at 1 mg per kg, we completely block the tumor growth. And on the water plots graph, you can see that we saw tumor reductions by between 50 to 80%. So we have a system which is working very well. We did only 2 injections. We can see tumor growth. We can block the tumor growth, but also see tumor reductions. And the treatment was very well tolerated. No modification in the toxical markers. We then ask if we can use the ADGN technology in combination with standard therapy. So we have combined ADGN 21 with Abraxane and ADGN 123 with capecitabine on different tumor models. 121 was treated on the PONC1 cells and xenographed mice when we have the G12D mutations. And on the graph, you can see that in the green is ADGN 121.5 mg per kg and Abraxane in yellow, 50 microgram. If we combine the two, we have a nice synergy, and we can see a tumor reduction by 80%. Similarly, with 123 in combination with capecitabine, in the green, it's just 123, capecitabine in yellow, and we combine in blue. We combine the two, and we have a very nice synergy. So this in vivo data suggests that we can use our ADGN technology in synergy with standard therapy. As I mentioned at the beginning, there's no drug on the market or approved by FDA which target G12D or G12V mutations of KRAS. 2 drugs have been approved to target KRAS G12C, which has sotorasib and agrabasib. But the limitation of those drugs is the emergence of resistance. So we have analyzed if we can use the ADGN technology, especially ADGN 122, which is target KRAS G12C, in the background when we have resistance to the currently used drugs. So for that, we select several cells which are resistant to sotorasib. The C1, C2 are just overexpressing KRAS G12C when you treat the cell with the sotorasib. And the two other clones got some resistant mutations. And what we show is whatever the cell line and whatever the mutation we got, the ADGN122 is still highly efficient on all the cells. And we have a very high IC50, around between 10 and 20 nanomolar, showing that we can use ADGN22 in the background when the cell or tumor are resistant to the currently drug, like sotorasib and adagrasib. So just to wrap up our data, so we have this peptide-based nanoparticle for the delivery of small size and large size oligonucleotide. We show that this technology can be used in vivo. We can use it for gene delivery and gene editing in vivo. We have a nice proof of concept that it works on different KRAS mutants. So we can do the editing of those different mutants. We show that we have a very nice tumor selectivity, that we can only need 2 doses, so 2 injections to block the tumor growth and see tumor regressions, and that the ADGN technology can be used in synergy with standard chemotherapy. In the context of resistance due to the use of small molecule inhibitor, we show that ADGN compound can overcome the resistance associated to the use of small molecule inhibitors. So in a wrap-up, this technology is very promising to target KRAS mutations in cancers. Just to conclude, I would like to acknowledge the team. As I said, all the R&D of that program is done in Nîmes, so that the team working in Nîmes at Diving Cells. The work is done in collaboration with the IAB and the Animal Facility Optimal in Grenoble. And we have a strong collaboration with GenPep for the chemistry. I thank you for your attention.